Thank you very much, Margaret. And I'd really like to thank all of you for coming on this Thursday. So we've had a really terrific talk from Hugh about heart diseases and some of the excellent work that Hugh and his colleagues have been doing around genetics and some of the treatment strategies they're putting in place for genetic heart disorders. So change of tone now. Really, I want to talk to you a little bit about what we're doing in leukemia, and in particular with respect to some of the work that the BRC here has supported over now some really 7 years, and also some work that has been supported over a much longer period of time by the University of Oxford and by the hospital and by the Medical Research Council in particular. So I just wanted to start out by a couple of quick definitions and really telling you a little bit about what leukemia actually is. So I think one thing that is central to everything we think about is really trying to understand how normal blood production occurs and how it goes wrong, because leukemia is a blood condition. And so one amazing important fact to remember when you're thinking about all of this is that every day we make about 10 billion new blood cells in our blood factory, which in adults is located in the bone marrow. And I can't think of a factory anywhere on earth where you're making 10 billion of anything every day perfectly. Well, the remarkable thing is not why does it go wrong. The remarkable thing really is why does it go right so often. So just bear that in mind. And I just thought I'd show you a picture of what a blood factory looks like. Many, many thanks, Hugh. Great. Is it that one there?
Yeah, that is the front one in the middle.
Yeah. There. Okay. So we take bone marrow out of the hip. It's a really big bone, and it's a very thin needle that we put in, and then we can just suck a little bit of the blood factory or bone marrow out. And if we spread it on a slide, it looks something like this. There are little bits on the end of it, and that's really what we focus on. And if we blow this little bit up here, you can see it looks like that. And there are these things that we call trails. And if we just focus on one of those trails there, it looks a bit like this. Now, you don't need to really understand this, but the important thing to see is that there are lots of different types of cells, lots and lots of different blood cells made every day, and that's healthy. However, this production can go wrong, and when we make too many blood cells in an uncontrolled fashion, it leads to cancer of blood cells, and I'll show you a little bit how that happens. So are blood cancers common? Well, they are. The most common cancers are set out here, and this is the number of cases per year, male, female. Most common, as you probably know, is breast, lung, prostate, bowel. But here are the blood cancers shown by arrows. And collectively, there are about 8,000 people a year in the UK diagnosed with leukemia, and they cause about 6,000 deaths. So it's one of the relatively smaller cancer groups, but it's a group nevertheless that causes a lot of unhappiness and sadness and death in families, and we see those families in Oxford, sadly. So the other thing about leukemia and blood cancers is that because blood cells are relatively easy to get, either simply from taking a blood sample through your arm or from the bone marrow, as I've showed you, we can get hold of blood cells very relatively straightforwardly, and because they're in suspension, they're all as single cells, as I've showed you. They're really easy to study, and that makes the study of blood cancers relatively easy. And often some of the findings we've made in blood cancers have been translated into cancers more generally. So when I first started thinking about this talk, the first thing I thought about is that, virtually every day or every week, we hear some good news story where people are talking about cancer being cured or blood cancer deaths being halved, or, there's a really good new cure coming up for cancer. And then, on the other hand, we hear stories like this where, we've already heard about Patrice Mumba, but here are a couple of other footballers who've been battling for their lives with cancer. And clearly, we all know this chap here, and he's been struggling with CLL in Cambridge for now many years. And slowly but surely, he's unfortunately getting a little bit more poorly each year. So really, where are we? Where are we with the myth and the hype, or where are we with the promise? So the first thing to say is leukemia isn't one condition. There are lots of conditions that we call leukemia. Some occur in children, some occur in adults. Some occur quite quickly, and the word acute really means that they're rapidly progressive, and there are different types of acute leukemia. I'm going to tell you a little bit later about acute myeloid leukemia, which is what I particularly see. And there's some that are much more slow-growing and indolent. And here are two types of chronic leukemia. And these can take many, many years to progress, and we have many treatments to slow them down, but eventually the leukemia overcomes the treatment line that a person is on. So the outcome is very variable, actually. And so when we're talking about curing cancer, we have to really think about a bit more detail. Do we mean children's leukemia? Do we mean adult leukemia? And what type of leukemia do we mean? So let's talk about a success story first, a nice positive way to start the talk, childhood acute lymphoid leukemia. This is the most common childhood cancer. And this is the survival of patients over the decades. And here I'm showing you a graph when in the 2000s, about 80% of children were surviving. Now in 2015, that's probably around 87, 88%. Brilliant. When treatment first started in the 1950s, the first doctors who treated patients were really pilloried in public. The doctors were thought of as being of the Joseph Mengele School of Medicine. Why were they trying to prolong the lives of these poor children who were destined to die? And it was very hard to do the first clinical studies, clinical trials in cancer medicine in children way back in the 1950s. However, since then, there have been a lot of clinical trials, and trials really mean studies where we take what we think is the best and we compare it with what we think is as good but could be better. And several chemotherapy agents were tried in combinations. The treatments for children usually last 1 or 2 years. Most of the time the child is at home, but the treatment is pretty arduous, and they're going to miss school, they're going to miss being with their friends. There are going to be times during those 1 or 2 years when they're in hospital and poorly. But the promise of those clinical trials has been delivered over the decades, and now most children are surviving. And the reason is large-scale clinical trials. These have been government-funded, and the acute leukemia trials that were done initially in the United Kingdom and then in America were renowned worldwide. And the structure of the NHS has really helped with this. I was in Montreal on Monday, and I was staggered to learn there are 10 hospitals in Montreal, and each hospital has its own complete different set of ethics for getting trials into place. Each hospital really doesn't do trials. There isn't a nationwide structure for doing clinical trials in leukemia in Canada. They're miles behind. And they really wanted to collaborate with us in the UK because they'd given up on their own regulatory processes back home. So I think the United Kingdom has a lot to thank the NHS, and it's got a lot to thank the national clinical trial structure that over decades the various governments have built up. Okay. So what about acute myeloid leukemia in adults? And here we have a real challenge. So let me show you what the challenge is. So this is the age of diagnosis of this particular leukemia, numbers of cases as a function of age. And unlike maybe some common myth around leukemia that it really happens in young people, most of acute myeloid leukemia happens in folks as they get older, and most over the age of 70. And there are about 2,500 cases, new cases of acute myeloid leukemia a year in the UK. So 80% of this are in the over 60 age group, and the median age of diagnosis is 78. So it's much more common in our aging population. And the treatment strategies are divided into those that are curative, where we really try and get rid of the tumor for good, and that involves quite aggressive chemotherapy and sometimes bone marrow transplantation. which is the only routine stem cell therapy that we have. And in the under 60s, this is the percent surviving over time. And you can see the curves flatten, which means that these people are going to live now forever. But it isn't great. It really isn't great. About, on average, half the patients survive, half the patients don't. And here I've shown 3 different curves, and we can use genetics that Hugh talked about to stratify patients into good risk, standard risk, and poor risk. But even the good risk patients, only about 2/3 survive. Now, in the over 60s, where we have most patients, the survival rates really haven't changed over the last 30 to 40 years. And this is where we need to make progress, and this is where we're finally making progress. And I'll tell you about that at the end. So what are we doing in Oxford about this? So there's been a really constructive partnership over the last few years between the university hospitals, the Oxford Biomedical Research Centre, and the university, really all focusing on clinical service, scientific programs, and from those 2 activities, really feeding into clinical trials that I talked to you about. Now, when a drug first comes to the clinic, we call it phase 1 clinical trials. They're very early clinical trials. And then when we have a bit of experience, we treat more patients in phase 2 trials and then ultimately in phase 3 trials. And the other key stakeholders in all of this have been patient support groups, and we've set up patient support groups in the Thames Valley, and also they're fed into national groups. The medical charities have been really outstanding, and the Leukemia Lymphoma Research Charity is one such charity. I went to Speaker's House last week when John Burko, together with Simon Stevens, the head of the NHS, and Jeremy Hunt were there, and they were really talking very positively about the impact that universities have made to the NHS and the charities have made. So again, that is a very wonderful thing the United Kingdom has. And we've worked closely and progressively with the pharmaceutical industries, not with a view to embellishing their profits, but with a view to getting new drugs into patients faster. So our catchment area is about 2.5 million people. We work with our partners in district general hospitals, and these are the kinds of numbers of patients and samples of marrows that we see per day and per year. Okay, so the people who've really made a big impact on the science in Oxford are set out here. And in particular, when I was a medical student, Professor David Weatherall, Sir David Weatherall, was the professor of medicine, and he was absolutely inspirational, and he brought, for many of us, genetics to medicine back in the 1980s. And his successor, Douglas Higgs, now runs the molecular hematology unit in this building called the Weatherall Institute of Medicine, which is part of Hugh's department, the Radcliffe Department of Medicine. And it really is one of the most important blood research units in Europe, and it's supported by a variety of people, as shown there. So let's just talk about now blood, and I'm going to tell you in the next few slides, the final few slides, some of the new treatments that are coming. So we've talked about blood production, we've talked about the number of cells in the bone marrow. There are about 10 to the 12, so that's 1,000 billion. And we know that there are 9 different blood types and that the bone marrow makes these cells for our entire life. But to make this production, you have to divide blood cells into distinct, what we call functional compartments. So there are maybe around 10,000 what we call stem cells in my bone marrow, in your bone marrows. These are very quiescent. They're mainly asleep. They don't really do much, but once in a while they come out, divide, and go back to sleep. But they're able to produce through their progeny all the blood cells that we make. So they're multipotent and they're very long-lived. They're with us for all our life. And they give rise, through division, cell division, to cells we call progenitors. There are many more, there are millions of these. They're still very long-lived, but now they make some choices. Maybe this one here is just going to make red cells, the one in red. And these then divide, make what we call later progenitors, and finally these mature into what we call terminally mature cells. So now these are in the blood. So these are now blood cells, these are what we call platelets, and these may be what we call white cells. And these cells, all of them, are destined to die. So this is like a conveyor belt. You start with a few cells, they multiply, 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 and as they multiply, they change in function till they finally reach the blood, and then they die. Okay, so this is a much more complicated diagram of that same process. Here are your stem cells. They renew a lot. These cells with the P behind them are your progenitors. They divide a lot, proliferate, and then finally you make these blood cells that come out into the blood. And in acute myeloid leukemia, what happens is that you have a virtual block in production so that you can't mature properly, and then all the cells behind continue dividing, and they dam up, and the cells really don't come out into the blood properly formed. And what's critical in this process is to understand what propagates the tumour. What cells here really are propagating that leukemia? And there have been two fundamental models, if you like, of cancer tumour propagation. One model, which is called the stochastic model, suggests that actually all the cells in the tumour are basically the same. All the cells are biological equivalent, and they exhibit variable behaviour either because they're in a particular place in the marrow, for example, for blood cancers, a place that really allows them to propagate tumour, or there may be external factors in the environment, like your hormones. They may be particularly receptive to a particular kind of hormone at a particular point in time. But the point here is that all of the cells can propagate the tumour. And the implication of that is you have to get rid of all of them to cure the tumour. That's a tall ask. But there may be some tumours like that. Now, the other model of the cancer propagation is what we call the stem cell model. And here, cells have stable but distinct functional abilities. So here, these purple cells are the only ones that can really propagate the tumour, whereas the others can't. And so to cure a tumour, you really have to get rid of these guys here. Now, acute myeloid leukemia has been held up as being an example of this. Now, I think these models are rather simplistic. I'm not going to get into the details of all of this, and they may change within a patient from the time they initially started treatment to the time when the tumour comes back. But by and large, we now have pretty good evidence that acute myeloid leukemia actually behaves this model more often than not. And that is really important because we now are targeting our treatments to these purple cells. So how do we go about purifying and identifying those myeloid leukemia stem cells? Let's call them that for now. Well, we take a bone marrow from a patient, and here is a marrow. It looks exactly like blood. We can then take it to a very complicated machine which will separate all the cells one by one in a stream and push them in different directions. And these machines nowadays can look at 10,000, 50,000 cells a second. So it's pretty amazing. And you can then separate out leukemic stem cells from all the other leukemia cells using these kinds of machines. They're called fax machines. And we then take these leukemic stem cells and we look for the telltale genetic changes. Now, most patients are not born with these genetic changes. They acquire them over the period of their life using sequencing machines. And Hugh talked a bit about DNA sequencing. Well, it turns out that because blood cells divide so frequently, every time a blood cell divides, you have to reproduce all that DNA. And that's 6 billion letters of DNA. And that's the book that tells cells how to behave. So if there are 10 billion cells dividing every day, and there are 6 billion characters of DNA that have to be replicated each time a cell divides, it's not hard to see how mistakes occur when DNA has to be completely replicated for the new cells that are being made. So mistakes happen. And if mistakes happen in blood genes, genes that really control blood production, this can progressively lead to leukemia. So we know now, and there's beautiful data that we and others have contributed to, that the first mutations in leukemia occur in normal stem cells. They expand the stem cell compartment, so these are acquisition of initiating mutations. And our first hints at this came from studying twins. And here are two girls, and they developed acute lymphoblastic leukemia. Here's a one of the twins who developed it, and this is her healthy twin. And what we realized is that the first mutations actually occurred in utero, in the fetus. And because these two twins were monozygotic, identical, and shared the same circulation through the same placenta, one of the leukemic cells passed from one twin to the other. So both of them had these initial pre-leukemic cells. But this twin here then developed additional genetic mutations and finally developed leukemia with multiple tumor propagating, here I'm showing 4 multiple stem cell populations, whereas this twin hasn't gone from here to here yet. And both the twins and their mum were incredibly brave because they were really, it's a really tough time for them, but they were very, very keen that the research that was done on this little girl's blood and this little girl's blood was made known to the public so people could understand how powerful this research is to understand risk and to understand how leukemia occurs. This girl had a bone marrow transplant and is now cured, thankfully. So this is how we understand leukemia today. Multiple propagating populations created from a normal stem cell that sets up what we call a pre-leukemic cell. Okay, so as a consequence of the Human Genome Project, which here is lots and lots of sequencing machines, this is how they looked way back in the day. Nowadays, they're becoming progressively smaller. And in Oxford, there's a fantastic company that now makes sequencing machines that are the size of a USB stick. Okay? And you can get a genome sequenced in a matter of days now. And there is a big program in the United Kingdom which has some merits, but it also has challenges, where they're sequencing 100,000 genomes to understand the genetic basis of many diseases, including rare diseases and cancers. So this technology is advancing at a remarkable rate, and the government is investing quite a lot of money into this. So when you find the gene, does it really make a difference? Well, it can do. So the poster child for targeted therapy came really through leukemia, and in particular a chronic type of leukemia called CML. And when they found the gene that caused CML, they were able to work out its crystal structure, which is what Hugh showed you when he was talking about molecular motifs in the heart. Here I'm showing you much more of a cartoon. This here is a gene that's mutated in CML. It is the causative genetic mutation. It's acquired usually, you're not born with it. And they were able to design a drug to particularly block that mutant protein. And this is what's happened to the survival curves. So back in the day, this is the percentage of patients responding to treatment, 10 to 20%. And since the start of imatinib treatment, that drug that specifically targets that mutation, we're now getting 95% survival. So this is what we all are working towards. But I think that for other leukemias and other cancers, it's going to be a little bit harder because there are many more genetic mutations per cell compared to CML. Now I'm going to end with this last slide. And here I'm going to tell you about a novel strategy that we've worked with collaborators in Stanford to try and really progress cancer treatment and leukemia treatment. But I think it's going to be relevant for many more cancers than just leukemia. So let me just tell you that, first of all, we all have cells in our body called phagocytic cells. And phagocytic cells remove unwanted cells by eating them up. And cancer cells, and probably most cancer cells, express pro-phagocytic or eat me signals. And we think they probably do that because they're damaged in some way. Now, we're not really sure why they do that, and there's quite a lot of work going on to understand that. But they do express pro-phagocytic signals. So here's a cancer cell expressing the eat me or pro-phagocytic signals. It'll be seen by a phagocyte, which is this grey thing here, and it'll eat the cancer cells. So you might ask, why do cancers ever develop? Well, they also express don't eat me signals. And here are the don't eat me signals shown in red. And these don't eat me signals are mediated by a molecule we call CD47. So here is CD47, 3 red blobs preventing the eat me signal and preventing the cancer cell being eaten. So together with Stanford, we developed a medicine that blocks the don't eat me signal. It's called an antibody. And now we're hoping that the cancer cells will be eaten. So we've done quite a lot of work over the last 7 years to show that that is a paradigm that certainly works in preclinical studies and works for numerous different types of cancers. So the clinical trial has now started, both in the University of Stanford and the University of Oxford. There's a trial in AML and myeloma. The AML trial is starting in June this year, and the solid tumor trial, which includes many different solid tumors, started in August in Stanford, August last year. We'll know, because AML is so aggressive and it's such a quick disease to take hold, we'll know probably by the end of this year, the beginning of next, whether this kind of treatment has any legs. And the reason why we like this kind of approach is because it's generic. It can be applied to many different types of tumors. The issue of targeted therapy, when you're looking at a particular gene and a particular mutation and targeting it in that way, is you're going to have to develop different drugs and most likely combinations of drugs for each individual patient. That's going to take quite a bit of time to work out. It's going to be potentially quite costly. One of the challenges the NHS will face, and you've heard about all of this in the newspapers, the Cancer Drugs Fund, for example, is how we can quickly test, effectively test new cancer treatments as they come, and how we're going to find out who the right patients are, who will respond to them, and how we're going to be able to afford this. So by using a slightly more generic approach, but an approach that targets tumor propagating cells, we hope to look at this problem in a slightly different way. Okay, I'd like to stop there and really want to thank you, and I'm very happy to answer any questions.